Answering MCAT questions in under 60 seconds. A certain eukaryotic organism contains multiple genes with highly similar sequences that encode proteins with similar functions, but these genes are expressed at different times in the organism's life cycle. The most likely mechanism by which these genes arose is what, right? So A says alternative splicing, right? Well, remember that alternative splicing is when you have a single gene, right, which is encoding a pre-mRNA, which is then spliced, right? And that pre-mRNA, due to basically varying types of splicing factors, can be spliced in different ways, such that different combination of exons are produced, and you get different mRNA transcripts encoding for different forms of a protein, like different, if it's an enzyme, like different isoenzymes and stuff like that, right? But those are all coming from a single gene. Here, they're talking about having multiple genes, which are basically variably expressed throughout the lifetime. Now, B, gene duplication, right? And notice that they're asking us how these genes arose, right? How they arose. Well, gene duplication is a mutational event where you literally duplicate a copy, you basically create a new copy of a gene, right? And through mutation events, you could potentially get multiple genes. And of course, since originally they came from the same gene, they might accumulate individual mutations throughout evolution, right? But they will encode probably pretty similar proteins. So gene duplication sounds like a really good bet here. Now, C, horizontal gene transfer. Well, that's usually when you have transfer of genetic material between two organisms, but oftentimes like in bacteria and something like that. But again, it wouldn't really explain why you have multiple genes with highly similar sequences. You know, generally these genes would be like between like unrelated organisms of some capacity with pretty variable genetic information, and it's pretty unlikely that you're going to be transferring genes that are very similar. Okay, gene duplication is a much better explanation. D, transcriptional regulation. Well, transcriptional regulation is specifically referring to perhaps why those genes are variably expressed throughout a lifetime, but it doesn't explain how the genes actually came to be. Why are they so similar to begin with, right? It's probably because they came from an original progenitor gene that was duplicated, and then the copies were mutated and underwent independent mutational events. But transcriptional regulation would be specifically referring to how why one gene is active and then it's silenced and then another one comes on, right? But gene duplication is explaining why these genes even came about. So B is the right answer.